All right. So, so I worked on a specific gene variant that sickle cell patients have, or just regular people have. Okay. So the objective of my research was um, to find an association between this XMNI polymorphism and a polymorphism is basically a SNP. So it's a single nucleotide polymorphism. Basically, one part of the gene is switched to another. So in this case, it's a C to a T. Okay, a C to a T is switched in this particular SNP. Okay, so it's in the promoter of that's actually it's in the gamma subunit. That's wrong. It's in the gamma subunit, and the presence of the SNP. So if you have a T right in this 158th position instead of a C, and you have sickle cell disease, it basically increases your fetal hemoglobin, right? And if you can't make a hemoglobin A, right? If you're making defective hemoglobin A, you have to rely on your fetal hemoglobin to kind of produce that oxygenation in the body. So this particular SNP has been well documented, and fetal hemoglobin modifying single nucleotide polymorphisms are associated with less severe disease. Okay, like people who have SNPs that increase hemoglobin F levels, they have less of acute chest syndrome. It's been documented, and they have usually less vasoocclusion. So these SNPs are very, very protective against people living with sickle cell disease. Okay, and if you have a high persistence of fetal hemoglobin, it's a phenomenon called HPFH. I don't know, maybe some people have heard it here. And so even though hemoglobin F levels, they've been, associated with less severe disease, there's really no correlation that's been found to predict with stroke risk. So it's still kind of a mystery why what's really happening with sickle cell disease and stroke and how it manifests in people. And so the objective of my study was to confirm an association between the XMNI SNP and hemoglobin F, and to see if there is an association with stroke, right? So for science, it has to be reproducible, right? There are researchers that have found this, and I said, let me see if I can reproduce this data and find that it's also significant. Okay, and so here's a chart that's kind of explaining the hemoglobin switches in regular people. Okay, so when an infant, right, is born, okay, let's start at the beginning. When a mother is pregnant, okay, that infant relies on fetal hemoglobin. That's why it's called fetal hemoglobin. They do not produce any hemoglobin A that adults produce. Okay, so but soon as that infant is born, or that fetus, we'll call it a fetus, as soon as that fetus is born, there's a switch that happens, right? So this is fetal hemoglobin right here. So fetal hemoglobin right here, when the baby is born, goes down, and the baby starts producing its own hemoglobin, right? We call that hemoglobin A, and this is the beta chain that's in hemoglobin A, right? So there's a switch that happens, and we don't understand this switch. Why the switch happens from why are we going from fetal hemoglobin to hemoglobin A? Okay, so one of my theories is that so one of the things that we know about fetal hemoglobin and regular hemoglobin that adults have is that fetal hemoglobin has high oxygen affinity. So it likes to bind to the oxygen more and not release it to the tissues. And this is probably because I think the fetus is really small, so it doesn't need a lot of oxygenation compared to an adult. So that's my theory as to why this switch happens. But genetically speaking, they're still trying to understand why this switch happens. Okay.